So thank you for having me here today. It's a pleasure to speak. And before we begin, I'd like to just give the disclosure that I'm co-founder and serve on the scientific advisory board of a company called Boundless Bio, and that's my only conflict. All right, so we do know that cancer is a disease of somatic DNA lesions. And one way to think about it might be that there's a sequence of mutations that progressively transform a cell into a cancerous state. But not all cancers follow this paradigm of a slow progression. And so I'm showing here this data from Steve Ellich's group, but there are many other papers like this, and they themselves were reanalyzing previously acquired data sets, where they show that somatic copy number amplifications, when they occur, they have very poor outcomes for patients. And these are cancers that are driven very quickly by the amplification of these oncogenes. And if you look at the picture out here, you can see that when the individuals have high number of mutations, their survival outcome turns out to be a little bit better than when they don't have a lot of mutations. But the inverse relationship holds when they have copy number amplifications, in that high copy number amplifications lead you to a very poor outcome. So a question that sort of really focus my lab is really what causes focal amplification in cancer. And a few years ago, we started a collaboration with Paul Michel's group, and we started looking at the different mechanisms that could lead to amplification. And so if you go back to the literature, there's work done by Barbara McClintock. She worked, of course, on maize, but the sort of sequence of breakage-fusion-bridge cycles that can lead to copy number amplifications has now also become a hallmark of cancer. And in fact, it's a textbook example of how amplifications can arise. And another sort of somewhat dated but very interesting suggestion was made that there are extrachromosomal DNA, or double minutes as they were previously known, that can also lead to amplification of copy number regions. And what I'm showing out here is work done by Jeff Wald's group back in the 80s. Okay, so in our collaboration with Paul Michel, we looked at almost 143 samples, close to 3,000 metaphase images, and we developed an image analysis tool that allows you to look at these images automatically and count extrachromosomal DNA elements if there are any. And in a paper that we published in 2017 and then subsequent functional assessment in 2019, we can see that there are indeed these circular elements. This is shown with an electron microscope. You can clearly see the circles and the double minutes being formed. Okay. And it turns out that these ecDNA are abundant in cancer. They were just sort of hiding in plain sight. So we looked at a number of cell lines as well as patient-derived xenografts, and you can look at the EC counts, and they range, of course, from small numbers to very, very large number, maybe hundreds of copies per cell. And their proportion in different cancer subtypes is also very high, ranging from almost 100% in, say, glioblastoma down to about 10-15% in colorectal tumor samples, right? So overall, we can see that ecDNA abound in cancers. Moreover, if you looked at the regions that were amplified by these, and you compare them to the amplified oncogenes in cancer, you can see that the most frequently amplified oncogenes all lie in regions that appear as extrachromosomal DNA, thereby suggesting that ecDNA are one of the main mechanisms by which oncogenes can be amplified. All right, so over the years, we've been working towards an ecDNA model of amplification. And the model is as follows. Fragments break off at random and form extrachromosomal elements. And replication initiation and recombination can lead to multiple copies. So there are many origins of replication, and each of these can replicate independently of the chromosome. But because they lack a centromere, when the cell divides, they don't need to divide equally. So you can see out here that the daughter cells might contain both replicated copies, and another daughter cell might not contain any copies. And if these elements, they are dysregulated, they don't bind to chromatin in the same way. And if they were to contain oncogenes, they would provide a strong proliferative signal. And so the presence of oncogenes then gives a selective advantage to the cells that carry. And because they preferentially divide, it leads to rapid copy number amplification in a non-Darwinian fashion. All right. And then finally, something that's somewhat controversial, but we see more of it, is that ecDNA might integrate back into chromosomal regions, and that might also explain focal amplifications. All right, so I don't, of course, have time to go through all of the different experiments that were done to validate tenets of this model. But what we are interested in is just looking at the structure of these extrachromosomal elements. Right? What's the fine structure? What are the genes and the regulatory elements that are carried on these ecDNA? All right, so a few years ago, Viraj Deshpande in my lab started looking at these structures just using next-gen sequencing, Illumina sequencing, and the tool he built, Amplicon Architect, was published last year. And it's a very, it's sort of simple to explain conceptually, even though it takes some time to build it. So we start by mapping all these short reads back to the reference, and we start with regions that you can see as being amplified with a high copy number. Right. And then we look at discordant reads where one end maps to this region, amplified region, and another end discordantly maps somewhere else, suggesting that these two regions are part of the same amplified structure. And so we collect these reads. We also look at regions where there's a sharp shift in coverage, because they are also illustrative of segments that have broken off, and breakpoints arise at these boundaries of coverage shifts. And then we look at additional breakpoint edges that explain these coverage shifts. And then finally, we have the sort of the outline of a structure. We do a more careful look at breakpoints. And the output of this structure is what we call a breakpoint graph, a graph that encodes all of the different possibilities of extrachromosomal or other amplified structures. And from this graph, we can pull out cycles. And the cycles, for example, might be illustrative of extrachromosomal DNA. And similarly, you can pull out paths. So Amplicon Architect over the years has become a very, very important tool in our lab. And here's one experiment that sort of illustrates its power. So here's a glioblastoma cell line, and it carries amplification of the epidermal growth factor receptor. You can see out here that there are hundreds of copies per cell in a simple cyclical structure that's extrachromosomal in origin, and multiple replicates give you the same structure. But when we apply erlotinib, which targets EGFR, the structure just disappears, and it sort of reintegrates back into the chromosome as a homogeneously staining region, showing up as a homogeneously staining region. On the other hand, other extrachromosomal elements containing different oncogenes, in this case MDM2, arise. And then when we remove the drug, the EGFR carrying ecDNA comes back up again, but now it has recombined with the other ecDNA, leading to a much more complex structure. So definitely Amplicon Architect has been an important tool which allows us to make these deductions just by looking at NGS data. But AA also has problems. So here's the example that we just covered is on the left. Here's another cell line, HCC827, which also amplifies EGFR with a copy number of 50, as you can see. But now if you take the naive cells and you have the structure and you treat it with erlotinib, there is no difference in the structure, right? So same experiment, same gene that's amplified. In one case, the application of the drug leads to a tremendous change in structure. The ecDNA disappears into HSR and then reappears with MDM2. In the other case, there's no change. Right? So what's happening? Well, if you look at the cytogenetic images, here's EGFR amplified, and you can also see CEP7, a centromeric probe or a pericentromeric probe. And you can see that in 827, the line that was not impacted by erlotinib, there's all these copies that are on the chromosomes. It's an intrachromosomal amplification. Whereas on the right side, if you look at the glioblastoma cell line where EGFR is also amplified, you can clearly see the ecDNA structures. And of course, these are all images taken from our earlier publications, right? So the cartoon really illustrates the challenge. When you have short reads, you can look at breakpoints and you can get a breakpoint graph, such as one that AA builds, but it doesn't distinguish between many alternative possibilities. So these two cyclical structures, they both satisfy all of the breakpoints, as well as the fact that segment A appears in copy number 3, whereas B, C, and D appear with copy number 1. But the orientation of B, C, and D relative to A could be different. On the other hand, if you had long reads that could span the structure A, then it could break that symmetry, because this long read, this single long read, can tell you that B is followed by A is followed by D, and therefore this structure where B and D are much further apart is not supported by this long read. Okay, so what we wanted to do therefore was to resolve some of these ambiguities in the breakpoint graph using long read technology, and what we decided to do was to use bio-nano optical maps. And the reason is very simple. These optical maps, once assembled, can be quite long, often 4 or 5 million base pairs in a few amplified cases, but with an average of 200 kb per fragment and close to 1 million base pairs on assembled reads. Right? So for those of you who may not be completely familiar with optical maps, here's a quick primer. What we do out here is not sequencing, but we look at positions of restriction sites. And so on this molecule, you can see all of these different restriction sites. And the only information of interest is the distance between different pairs of restriction sites. And that distance, if it is matched up with other molecules, you can use that to assemble the two molecules together. Or you can map an optical map to a specific region on the genome where these restriction sites have been mapped in silico, just looking at the positions where these restriction site patterns appear. And so you can do mapping and scaffolding using optical maps, if not full assembly. And so what we do in Amplicon Reconstructor is that we start with an AA-derived breakpoint graph. Although you don't have to run AA, you could have your own breakpoint graph established using short reads. And we also take assembled optical map contigs. We're using the ones provided by the bio-nano assembler. And then AR runs on these and produces reconstructed amplicons, elucidations of what the amplicon structure might look like. Right? So here's sort of the heart of the algorithm in detail. We start with these segments, and these are connected by breakpoint edges. And so this is a breakpoint graph output by Amplicon Architect. We take each of these segments and produce an in silico optical map by mapping the positions of restriction sites. And then we take the assembled OM contigs, the optical map contigs, and map these to the fragments, align them to the fragments. We use our own tool called SegAligner, and we have now better versions of this tool. And this allows us to create scaffolds. Scaffolds where a single optical map contig is aligned with multiple segments, and the segments can be ordered and oriented based on the contig information. So that scaffold already provides a lot of information. But we also take smaller fragments that have very few sites that couldn't previously be mapped unambiguously, but we can find paths in the breakpoint graph that connect these small segments in a way that is consistent with the sites seen on the scaffold. So the scaffold also allows us to impute these paths and lead into a much richer and carefully elucidated structure. And then finally, we take segments that can connect multiple contigs together and chain those segments, and that leads us to the final path. Okay, so the input again is breakpoint graph and optical map contigs, and the output are these complicated structures of amplicons, which can be millions of base pairs. And then by looking at these paths and cycles, we can begin to get at what the Amplicon structure might be, and more importantly, what's the mechanism that might have created these Amplicon structures. Right? So the AR output looks, we have our own sort of visualization tools, and you can see out here that we either have a linear view, which many of you might have been sort of have looked at before, where we align the genomic segments with their in silico digestions against optical map contigs. And these could be chains of many optical map contigs. But we also provide a circular view, which makes more sense for extrachromosomal DNA. And so the outermost circle gives sort of the genomic annotation. Here are the genes and the exons of the genes on the outermost circle. And then the middle path is, the middle circle is the in silico digestion of the genomic region. And the innermost is the optical map contig. In this case, you can see that a single optical map contig provides evidence for this circular structure. Okay, so we first started by testing Amplicon Reconstructor on simulations because there are not many gold standard data sets available where the mechanism of amplification is well known. So we took optical maps, and we had previous cancer samples which we had studied using both cytogenetics and Amplicon Architect, and we had some idea of what the structure looked like. And we took the sort of long paths and cycles which corresponded to Amplicon structures, and we sort of simulated OM molecules from these paths, and then just to mimic the entire process, we assembled them using the BioNano assembler, and then these were considered to be the optical map contigs. We mix them with optical maps from the entire genome. So we have sort of whole genome optical map contig information. And we had sort of 85 of these genomes, which have focal amplifications that were based on real data. And you can see that the results were very reasonable. On the x-axis here shows the proportion of samples. And then the y-axis shows both precision and recall, using a variety of measures. Here we are using what we call the length measure. So the recall suggests that you may have a certain proportion of the fragments that were reconstructed using Amplicon Architect. And so in 80% of the cases, or 90% of the cases, we have close to 80% recall and close to 85% precision, as you can see in this picture. Okay, so there's a lot more data that we provide in our publication, and I encourage you to look at it. It's on bioRxiv, and hopefully it'll be published in the near future. All right, so we had, now that we had our tool, we decided to apply it on cell lines that have previously shown very complex rearrangements, and here's sort of a listing of the cell lines. And as we walk through some of these examples, I will talk about how these cell lines are different in their forms of amplification. All right, so let's start with the simple glioblastoma cell line. It's a brain cancer, and when we ran Amplicon Architect, you can see that there's a very simple cycle with 3 important breakpoints. Breakpoint 2 is the EGFR V3 deletion, and then breakpoint 3 is a small sort of deletion in the middle of this region. Breakpoint 1 connects forward to backward, completing the cycle. And then when we ran it through AR, we got a single contig that completely explains the circular structure. So this is one of those easy cases. You can see the 3 breakpoints, 1, 2, and 3 in the cyclic view. And a single config in sort of the innermost circle that suggests what the fine structure of this amplicon might be. Right? And it's about 1.2 million base pairs in length. Okay. On to a more complex structure. So K562 is a CML breakline, a leukemia breakline. And it has, you can see it has a very complex structure. The most interesting thing that people observe is the fusion between chromosome 9 and 22, which forms the fusion Bcr-abl product, and that causes the pathogenicity. And the Amplicon Architect structure in this case was quite complex. But most interestingly, it had not only chromosome 9 and 22 with the Bcr-abl fusion, but was also connected with a portion of chromosome 3 with another gene, GPC5. And so the structure at least did not appear to be a simple translocation, as is posited in many studies. So when we reconstruct it using Amplicon Reconstructor, we get a scaffold with four optical map contexts, 5 optical map contexts that cover close to 8 million base pairs. And you can see the Bcr-abl fusion out here between chromosome 9 and 22, but also a very clear connection with GPC5 on chromosome 13. Okay, so this is one example where the AR structure very nicely resolves a much more complex structure graph that Amplicon Architect had created. To cytogenetically validate, we probed this using Bcr-abl GPC5, and you can see from these slides that they all map to the same location. And so here's on the very right is the combined set of images. And on the bottom panels are the amplified versions of these images, suggesting that not only was there a translocation, but there's also a very complicated structure to an amplification of these regions. And what might also be interesting is that this region is very small. It might be intrachromosomal, but it may also be an extrachromosomal structure that doesn't contain a centromere, and that remains to be elucidated. So we are also then looked at next the lung cancer cell line that we talked about before, where we had EGFR amplified intrachromosomally, and therefore, erlotinib treatment did not change anything in the structure. This cell line is previously posited to have the breakage-fusion-bridge as a mechanism of amplification. That's what causes this intrachromosomal amplification. And really what's happening out here, for those of you who may not be familiar with these breakage-fusion-bridge cycles, is that a telomere breaks off. And when the telomere breaks off and the sister chromatids are formed, then there's a fusion that takes place to stabilize the chromosome. And when the centromeres centromeres move apart during cell division, it breaks again, but the break may not happen where the fusion took place, and that results in the inversion and duplication of a large chunk of fragments. So here you can see that this ABCD fragment, which can be quite large, is both replicated and inverted in the rearranged structure. And then the cycle can continue on in subsequent cell divisions, leading to tremendous rearrangement and amplification of a large number of regions. Okay, so you can see that the architecture of this would be very complex. When we ran this structure, the whole genome sequence through Amplicon Architect, we could see these segments A, B, C, D, and E together close to about 9 million base pairs with this characteristic step-like rearrangement. And that led to sort of our understanding that this could be breakage-fusion-bridge, but it did not have the power to sort of fully elucidate the structure. Okay. So when we run Amplicon Reconstructor on it using bio-nano reads, you can see that we get 5 amplicons that clearly show these fold back operations. So you can see that this B minus A plus is inverted and replicated in a 2 million base pair scaffold. There's another 1.4 million base pair scaffold with this C plus D minus inverted and duplicated, and so on, all these 3. And then when we combine all of these information, we can get a very clear breakage fusion structure, which is shown at the bottom here. And we could sort of manually reconstruct a series of operations of prefix inversions and duplications that could lead from this A, B, C, D, E, a single structure, down to the more complicated breakage-fusion-bridge amplicon at the bottom. And besides these prefix inversion and duplication operations, there was also some local rearrangements and deletions that took place along the way. And on the left side, you can see this operation in sort of a schematic fashion, which shows how these fold-back reads are creating the structure. And finally, this entire structure, this 9 million base pair structure, also repeats and makes many, many copies of itself, leading to this tremendous intrachromosomal amplification. All right, so we also did some cytogenetic validation, taking probes for each of these segments, segments A, C, and D, which are interleaved. And clearly the cytogenetics shows that these probes are indeed interleaved and appear in multiple copies on the chromosome. All right, finally, we go back to a few cell lines which had extrachromosomal DNA, so seemed relatively simple, but they also showed up with intrachromosomal or HSR looking events. So the same, here's one cell with extrachromosomal DNA, here's another cell in metaphase with the same structure apparently on the chromosomes. And so when we elucidate this using Amplicon Architect and Amplicon Reconstructor, we do get a very clear circular structure with 3 optical map contigs scaffolded together, and suggesting that indeed the same cyclic structure is found in both extrachromosomal DNA and HSR, suggesting that the ecDNA formed and then reintegrated back into the chromosome. We also looked at NCI H460, another glioblastoma cell line, this time with the MYC amplification. And we get a linear structure, but this has this very nice property that suggests that it was extrachromosomal and circular in origin. You can see that this MYC and PVT from sort of 11 o'clock to 1 o'clock is completely duplicated out here between 3 and 5 o'clock, and also duplicated and inverted between 1 and 3 o'clock. And so you can see that the same MYC PVT containing segment appears 3 times in this structure. And so if we sort of look at this data, we can sort of postulate that this works, this is consistent with this agglomerative model of ecDNA based amplification, where different circular ecDNA elements might recombine to form larger structures, which could then lead to integration into a genomic location. So in summary, Amplicon Architect and Amplicon Reconstructor together allow us to first, to get at the fine structure of the amplifications. Especially Amplicon Reconstructor helps sort of elucidate all the ambiguities that might be present in the Amplicon Architect reconstructed breakpoint graph. And it also hints at, if not completely proves, mechanisms of amplification. So we looked at many cell lines, including chromosomal translocations, extrachromosomal DNA formation, breakage-fusion-bridge formation, and the AR structures unambiguously could point to one of these mechanisms as being the important mechanism of amplification. Okay, so we still have some questions though. For example, where do these ecDNA reintegrate, right? So again, if we had a short read, it would be very difficult to figure out where this reintegration happened, because it would not appear in the same large copy number that these amplified structures appear. But if you have a long read as provided by BioNano, we can see that this long read can allow us to get at integration points. But in order to elucidate these points of integration, we would need to work with individual fragments, not assembled bio-nanomolecules. So in some sense, we have to go back to the drawing board, and our more recent work now really hints at trying to look at these discordantly mapping single optical map fragments, which are a lot more error prone than assembled bio-nano contigs, but they still contain enough signal to be able to find these integration points. And of course, our data is very preliminary, but I wanted to show you some examples. We built our own fragment mapping tool, and we compared it with some of the existing fragment mapping tools, one of them being OM-BLAST. And this is unpublished data from our lab, which shows that at least in terms of time, we are close to twice as fast as OM blast, sometimes 3 times as fast, and maintain very high sensitivity. Each of these, numbers from 1 to 6 and the x-axis represent different samples. So in some cases, our sensitivity is very, very high. And in every case, it's at least as good as OM blast. All right, so we have this sort of preliminary tool, and we can use it to look for points of integration. And in H460, which was this complex ecDNA that reintegrated, we can see that the structure actually integrates into chromosome 12. It's an EGFR containing structure on chromosome 8 that integrates into chromosome 12. I'm sorry, it's a MYC containing amplification that reintegrates into chromosome 12. And if you look at K562, we also find this discordant map that connects it with the chromosome 13 amplicon with chromosome 1. So again, this needs to be proven, but it suggests that not only can we find defined structure, these long reads also can help us look to see where these amplified structures might be reintegrating back into chromosomes. Okay. So with that, I'd like to sort of conclude our talk by summarizing and saying that a combination of next-gen sequencing short reads and the long optical map contigs can provide us for the first time an integrated look at large amplified structures in cancer and allow us to get at mechanisms. A lot of people helped me with this project. Jens Lubeck, his work, though is still unpublished, is sort of, he did the main work on Amplicon Reconstructor, and most of the work that I talked about today was work done by him. Viraj Deshpande in my lab, he's graduated, and he built the tool Amplicon Architect. And then Siavash Raesi builds, along with Jens Lubeck, build Fandom, which allows us to look at single optical map molecule fragments and map them back to the reference. Right, and we also want to acknowledge great support from the Michel lab, especially Kristen Turner and Sean Wu, but many others in the lab who helped us both with the cytogenetics and with interpretation of the structures. And then finally, BioNano, David Pai, and Alex Hasty were very, very helpful in presenting us both with their data as well as helping us interpret some of our results. Okay, and finally, I'd like to acknowledge funding from the NIH and NSF. Thank you for listening.